Okay. Here we are. Okay, so exam 1 grades, about half of you have them back. The other half of you, I'm hoping to get them to ASAP. I was hoping for today, but hopefully tonight, late tonight, maybe tomorrow. It all depends on a few outside of my control things. Okay. Take 5 minutes to write. Signaling through GPCRs usually involves production or release of intracellular signaling molecules known as second messengers. In one example in class, signaling resulted in activation of adenyl cyclase. What second messenger is produced, and how does it activate gene expression? Okay, so a question I just got is, are you talking about cyclic AMP? And I'm like, yeah. So for those of you who are like, what the hell is she talking about? I see a lot of people going like
No, I just think yeah.
Okay. So how about we see what Joseph Tobin, who's usually in the front, is over here. You know today is the first day of Sukkot. I don't know. Who knows what Sukkot is? A Jewish thing. It's a Jewish thing. It's a Jewish holiday, so some people aren't here. I don't know if Joe Tobin is at Sukkot. Okay. Have you guys all seen? There's a sukkah up by Union. Pond. Okay. Go look. It's outside. It's like a little hut you can eat in. Okay. Meta Schwartz. So we already came up with cyclic AMP. I really want you guys, because we always learn about this small molecule release. So what does it do?
So is adenyl cyclase like cyclic AMP in this case?
It's the enzyme that makes
Okay.
cyclic AMP.
Okay, so cyclic AMP, I wrote binds to and activates PKA, which is a cyclic AMP dependent protein kinase, and then that PKA then enters the nucleus.
Right.
So activate.
Right, exactly. So I always want you guys to be thinking about, well, what is the second messenger doing? What is it actually doing? And exactly. So so signaling through the GPCR that we talked about on Tuesday activates adenyl cyclase, which is the enzyme that makes cyclic AMP. If you think about the names, adenyl cyclase, it's makes things cyclic. Cyclic AMP. Exactly. So that's a second messenger. Second messengers are these small molecules that kind of help to relay the signal from the G protein or from the receptor to the rest of the signaling pathway. And so what it's doing here is it's binding to PKA and activating it. And then that kinase can go into the nucleus and phosphorylate things to promote gene expression.
Say that again. So it activates PKA and
You said PKA, right? It activates PKA. PKA is a kinase, so that means it's going to go phosphorylate something, right? So it goes into the nucleus and phosphorylates gene regulatory proteins to promote gene expression. Yes.
I have down that it activates CREB, but does it activate multiple things?
Well, so Krebs is down the road, right? So so what cyclic AMP does is it physically binds to PKA and releases an inhibitor. Then PKA can go and phosphorylate Krebs. Phosphorylate Krebs and activate gene expression
Does
mass.
it CREB though, or does it?
A lot of different things, but in what we spoke about in class, we spoke about Krebs. Yeah.
I'm kind of confused. Because I remember in AMP, I learned that the second messenger was a G protein. So
You learned that the second messenger was a G protein.
like the G protein was the second messenger.
It activates. It's an activator of the G protein itself is a G protein. The second messenger is the thing that's being released. So calcium release. You probably learned about calcium release in AMP, right? Muscle contraction, all that. So there you're having release of calcium. Second messenger. A lot of the time, the signaling activates the G protein. That's going to activate whatever enzyme is going to result in the release or production of that second messenger. So you must have learned it in a very basic way in AMP. Because maybe had you gotten the whole pathway, you would have started crying. I don't know. But you were right that whoever taught you was right in that G proteins activate.
But the G protein is not
The G protein is not a second messenger itself. No. No. Who thought G proteins were second messengers? Okay. So at least it's not a really massive. Because I had the people think that you have to replicate your DNA before transcription and translation. And I remember taking a vote in class, and there was something like 50% of the class had been taught in a way that they believed you needed to copy your DNA before transcription. Who thinks that you need to copy your DNA before transcription? Well, now you know that you're not supposed to raise your hand. But back then, 50% of the class thought that. So sometimes there's just the way we teach things creates misconceptions. Okay. Good. Any questions about this? Okay, so when we're learning about signaling, I always want you to be asking yourself, well, what is this doing to promote the end result? Okay, so far, what have we done? We've been talking about signaling. We introduced a few example pathways. We talked about different types of signaling molecules, different types of receptors, and then we started talking more specifically about how GPCRs work. And we also introduced some general mechanisms for turning off signaling. One of which you guys are talking about in replication this week, right? But it's not turning off signaling, it's actually turning on signaling. Woo. Crazy. Okay. So we were talking about cyclic AMP last time, right? And GPCR. So here's another second. We're going to talk about another second messenger. So second messenger number 1 related to GPCRs, for those of you keeping an outline, was cyclic AMP. Second messenger number 2 in relation to GPCRs is going to be calcium. So some GPCRs, so remember all today's not a good day for me. All GPCRs activate some other enzyme, right? So with the cyclic AMP example, it was activating adenylyl cyclase. So with calcium, it's activating phospholipase C beta. A phospholipase. It could be a mouthful or not. Who thinks phospholipase is a mouthful? Who thinks phospholipase C beta is more of a mouthful? Okay, so let's go with a smaller mouthful. So it activates a phospholipase. What does that mean? What do you think a phospholipase does? Just based on the word phospholipase. So I'm hearing it phosphorylates something. Well, kinases are what phosphorylate. So it's a phosphorylated enzyme that would do something to the lipid. Well, definitely will do something to the lipid, and it has to do with a phosphate group. Well, what it's doing, yeah.
Does it cleave all phosphates from fats?
Yes, it cleaves off phosphates from fats. So it's kind of all in the word. I think a lot of the time, so you can always assume ACE means enzyme, but a lot of the time, like lipase, protease, nuclease, you're talking about degradation of something, right? So, or cleavage of something. So phospholipase is cleavage of some phosphate group from some lipid. And in this case, your lipid's in your plasma membrane. And so this figure is showing the gray box is supposed to be your inner monolayer of your lipid bilayer on your cell, your plasma membrane, and it contains some of this phospholipid, which is called either PI45 bisphosphate, or some people call it PIP2. Easier. And so you could see this is your fatty acid, right? And then you've got phosphate groups and this ring here. So basically what's happening is you're getting cleavage, and that leaves you with this bit, which is diacylglycerol, and this bit, which is IP3. So basically it's cleaving off phosphates from your lipid. And so that's going to do something to activate signaling. There's going to be something that happens here. This creates a binding site for something. And then this is going to bind to something too. What does it do? So this is showing it in more of a signaling pathway figure, where you've got your signaling through your GPCR and activated G protein, which is actually what we have been talking about. That's consistent among all GPCRs. Signaling molecule activates your GPCR, conformational change, kicks off GDP, binds GTP, that's all pretty standard. And then this G protein is going to go activate something. Last time it was adenyl cyclase to make cyclic AMP, but this time it's phospholipase C beta. And it's going to cleave, so you end up with a diacylglycerol and IP3. Did I say that last time? I was right. Okay. So diacylglycerol, that makes a binding site for protein kinase C. And so it comes up there and binds to that diacylglycerol. And then IP3 is released, and it can go bind to what kind of receptor is this? Ion channel receptor. And it can bind to this ion channel receptor and open it and release calcium from the ER. Now you have to ask yourself, okay, so what's the second messenger here? It's calcium. This whole thing, what it's doing, it's exactly the same as cyclic AMP, right? It's the same exact theme. Activation of an enzyme that results in release of, or production, here's release of a second messenger before you produce cyclic AMP, and here you're releasing calcium. And what is that going to do? It's going to go bind to something and activate it. And so here it's going to bind to that protein kinase C, PKC, and it's going to activate it. And so now you have an active kinase, just like you had PKA that became activated and went on to phosphorylate things and activate gene expression. This is going to become active, go on and phosphorylate things and activate the signaling pathways. Okay? So these are very similar. I want you to think about the similarities. You can either look at these as like, oh my god, there's 10 steps, I don't know what's going on. Or you can think of it in an organized way of like, these parts are all the same all the way down, and these things have analogous functions all the way down, and then it starts to make sense because your end goal here is pretty much activation of a kinase that's going to promote the rest of the signaling pathway. Okay? Yes?
So is the IP3 not considered a second messenger?
Is the IP3 not considered a second messenger? I don't know. I think maybe it is, because it is something that's being released and going to activate something, right? I don't think of it as a second messenger, but I kind of feel like it's acting the same way. We'll have to look it up and see if it's thought of that way. Okay, any other questions? Good. Yes.
Is the IP3 that in Citron?
Yeah.
Okay.
Yeah, so we've got the big name and then the easier to remember name. Okay. So you gotta think to yourself, okay, so if calcium is activating these kinases, then there's gotta be some mechanism for keeping calcium low normally in the cell, right? In the cytoplasm. And so they're kept low through two major mechanisms. One are these calcium pumps or calcium exchangers that are constantly pumping calcium out of the cell, so out of the cytosol. So calcium pumps in the plasma membrane, or into the ER from the cytosol, so still out of the cytosol, or into the mitochondrium. And then there are also calcium binding proteins that are not PKC, just binding proteins, and their role is to bind to and sequester calcium. So it's not available for binding and activating PKC, right? Because if you have a lot of calcium in the cytosol, well, what's the point of having these inducible mechanisms for activating PKC, right? Because then it would always be binding and always have. So a nice exam question could be, a mutation in your calcium pump would have the following effect on signaling or on whatever pathway. And you would have to think to yourself, okay, well the calcium pump, it's not working, so that means there's high cytosolic calcium, so there must be a lot of calcium that can bind to PKC, so you've got an active kinase, so the different signaling pathways that are controlled by that kinase must be constitutively active. That's the way you would work through that. Any questions? Okay, so cyclic AMP and calcium are two examples of intracellular mediators. That's what the textbook calls it, these intracellular mediators. Other people call them second messengers, same thing. That activate other enzymes, right? In both cases here, they're activating kinases that relay the message affecting gene expression and or other effects on cell function. Right? So this is all about signaling pathways. And it's all about relaying a message from outside of the cell to somewhere else within the cell. So the cell can respond to something. So as with anything in the cell, if you turn it on, you need a way to turn it off. And so with GPCRs, we call that desensitization. And it depends on phosphorylation of the GPCR and then recruitment of arrestin. And so what's interesting is the phosphorylation occurs by that kinase that was just activated. So like PKA. That kinase becomes activated. It's not only going to go activate gene expression, but it's also going to go up to the receptor and phosphorylate it. And that so this is the GPCR kinase. So there's GRK also that will do this, but then they also use other kinases that were just activated, come up, and they phosphorylate the receptor. And that results in creation of a binding site for recruitment of arrestin. So what do you think the outcome is of recruitment of arrestin, other than the fact that it says it in the word? What do you think? What do you think is happening to this receptor? Is it active or not? Not, yes. Sometimes I just need something ask the direct question. Right, okay, it's not, but why, and so why isn't it active? Other than the fact that arrestin is binding. What part of GPCR signaling is being inhibited? Well, phosphorylation is happening, right? But what part, think about how a GPCR works and what happens when a ligand binds. And so when you have phosphorylation here and binding of arrestin here, what part of GPCR signaling can't happen there?
Is it
Well, the binding of the ligand can still happen, and the conformational change can still happen, but normally what does that conformational change do? From what? From the receptor or from the G protein? Right. So you notice there's no G protein in any of these pictures, just the receptors there. Because remember, this is a two-part thing. You've got your G protein. It's a G protein coupled receptor. It's not a receptor that is a G protein. They're two different proteins that work together. And so, actually, protein complexes. So if you've got your receptor bound to a ligand and then you recruit this big protein here, it can no longer interact with the G protein. And so you can no longer activate your G protein. So you were there, you were so close. You were like right on the verge. I saw a hand going up. You? No. So, how does the kinase know when to desensitize the cell? How come this? So how does the cell know that it's time for phosphorylation to happen and for arrestin to get recruited? Does it just randomly happen, or is it a regulated thing? Obviously, thank you. How? So I just kind of told you. There's some form of negative feedback kind of going on here, right? Who can remember what that negative feedback was? Right, the kinase that was being activated is the same one that's going to phosphorylate the receptor. And once that receptor is phosphorylated, you'll get recruitment of arrestin. So what happens if you have a constitutively active GPCR kinase? So this blue guy is always on. So you'll always have binding of arrestin. What else? Go further than that. The GPCR will be off all the time. I feel like I've lost everyone. Did I lose everyone? Who's like, I wish I was still learning about vesicular transport. Yeah. How does the kinase know when to desensitize the cell? Yeah. So you need to have an activated kinase, and some of the kinases that will phosphorylate the receptor are the kinases that are activated by the receptor. So it's negative feedback. So these receptors activate PKA and PKC, right, through binding of the second messengers. As soon as they're active, they'll go on and promote signaling, but they'll also come up and phosphorylate the receptor to turn it on. Okay, so I want you to think of another thing. Now you have to think about vesicular transport again. What is the role of arrestin in endocytosis? Just guess. Or do you think it plays any role in endocytosis? Or do you think that part of cell biology is over, so this part will have nothing to do with it? Do you think there's a way that arrestin might be interacting with the endocytic machinery? Yeah, because I'm asking that. Yeah. Exactly. It interacts with clathrin. It acts like adaptive. Right? And so it interacts with clathrin. And so not only are you going to get shut off of signaling through inhibition of interaction with your G protein, but you'll also get shut off through endocytosis. So two roles for arrestin in GPCR sensitization. Desensitization. Good. Someone needs to send me back to sleep. Yes. Yes. Okay. I thought clathrin was involved in formation of vesicles. What does that have to do with endocytosis? I'm going to have someone else answer. Who wants to answer that one? It's fresh in your mind, right? For those who didn't do braids on, yes. I'm talking about just endocytosis. So what does clathrin have to do with endocytosis? Yep.
So, clathrin is used actually for the vessel.
Clathrin is used to form the vesicle. Right? So, or a vesicle that's formed through endocytosis is called an endosome, right?
Yeah.
So that's the protein coat that's required for endosome formation. Yes?
Does adaptin bind to elastin?
Adapt. So you can think of adaptin and arrestin as synonymous in function.
Okay. So the clathrin bind to elastin.
Yeah. Yeah. Okay, so who's confused by this merging of topics? Raise your hand if you're confused. Thank you. Because then I'll try to spend another minute. Okay. Right, so remember when we learned about endocytosis and we learned about receptors being endocytosis and clathrin-coated pits and endosome formation, and then the endosome acidifies and then goes to lysosome. Yes. Kind of lost, yeah.
So you said arrestin acts like that.
Arrestin is synonymous in function.
That's where I lost it.
Huh?
So I know that arrestin acts like that, and then
How's adaptin linked with clathrin?
that
Huh?
clathrin is needed. Adaptin's needed.
Right, yeah. Adaptin is what links the receptor for cargo or the cell surface receptor to clathrin so that you can start forming vessels.
Will it downregulate the GPCR? Will it downregulate?
Yeah, it would downregulate the GPCR. Sometimes it's hard to make these connections, so that's why I'm going to spend a minute or two. Okay, so I'm hearing over here, let's avoid side conversations. If any of you guys have a better way to explain it,
I was trying to hear.
okay. Do you have a better way to explain it?
No, I was gonna make sure I was understanding.
Okay, so you reiterate, then you next. Okay.
You were just saying that arrestin acts as adaptin in interaction with clathrin, but as an inhibitor instead of activating.
No.
Okay.
It's an inhibitor of signaling because you're downregulating a receptor. So remember we spoke about endocytosis as a form of receptor downregulation, right? And recruitment of receptors into clathrin-coated pits. That's what this is.
I got that part, but what my question is, does that mean that every vesicle with clathrin coat is destined for endocytosis?
No. So the question was, is every vesicle with a clathrin coat destined for endocytosis? No, because clathrin coat is used for not just endocytosis.
Exactly, so that's what I'm,
But every vesicle with arrestin, every time arrestin is recruited, so every time clathrin is recruited to the plasma membrane, that vesicle that forms will become an endosome. That encourages budding inwards or endocytosis. Just like all other protein codes encourage budding, right? Hop 1, hop 2. The whole purpose of it is to encourage budding.
so all vesicles with clathrin coat are destined to become endosomes, therefore.
All vesicles with a coat, all membranes that have a coat recruited are destined to become a vesicle. Every time clathrin is recruited to the plasma membrane, you'll get endocytosis. Yeah. So what happens when the GPCR interacts with clathrin? What happens? So what happens when clathrin is recruited to the plasma membrane? Right. So remember we talked about receptor downregulation and recruitment of receptors into clathrin-coated pits with adaptin and clathrin, remember, and then you get endocytosis. This is the same exact thing except for the fact the receptor is a G protein coupled receptor. It's not a little Y like I was drawing. But it's going to be recruited into clathrin-coated pits and be endocytosed and downregulated. Because remember, we're talking about desensitization. Desensitization means turning off signaling. Right? So a good way to turn off signaling is to send your receptor to be degraded or to sequester it. Right? Okay. Is anyone still horribly lost?
So
Yes.
it does get degraded?
I don't want to say, I never like to say always something happens, but getting degraded is an option. And just like, remember, with endocytosis, you're not always degraded, right? But unless diverted elsewhere, the route should be to the lysosome. Yes?
Do you need a multivesicular body for this, or no, because it's attached to a
So do you think it would butt into multivesicular bodies? Is the question. It seems logical because you usually want to hide signaling, but at the same time, because you've got arrestin bound, there's not going to be any signaling on the cytoplasmic side. So it's not like there's this cytoplasmic tail with all these signaling proteins bound to it that are going to continue signaling, because you have this inhibitor bound. Yes?
So I'm still kind of confused about like what the rest of the systems here. Is it kind of helping? It's helping facilitate the endocytosis?
So if I was to cross out the word arrestin and rename it adaptive, would it make sense?
So
So it's the same thing. Yep.
So it's just facilitating endocytosis.
Yep. Yep.
So it's the GPCR that's inside the endosome for degradation, right?
The GPCR will be inside the endosome, will it be degraded? Unless diverted elsewhere, like a recycling endosome, it should be degraded. Yep. Yep. Remember the end result here, right? Downregulation. Okay, are we feeling better? Everyone happy?
Sorry,
No.
one more question.
I love questions.
Arrestin, though, it really is the same thing as adaptin? Adaptin will, as soon as it makes the vesicle, it will be taken off from the naked vesicle. But here, it's also being used to downregulate the signaling. So it does.
It interacts with clathrin. So the question is arrestin really just like adaptin? And what we would have to do, the real way to answer this question would be to look on NCBI gene or somewhere with the protein sequence and see if there's a crystal structure and start to see what domains are here. I can guarantee there's some sort of phospho something binding domain on this side, and there's probably some sort of clathrin binding something side on that side. But would it be released just like adaptin is released right away? I would guess so, because that's how it tends to be, but I don't know with the rest. I know clathrin should be released.
Yeah,
But I don't know if arrestin would be, because maybe it needs to interact to inhibit signaling, or maybe that it needs to go into multivesicular bodies. I don't know that level of detail, but it could be easy to look up. Do we want to do Stump the Teacher?
Yes.
Okay. Need more work for me. Okay. So the question is arrestin, so since arrestin participates in endocytosis of GPCRs, what happens then is how is signaling kept off? Does arrestin remain bound to the GPCR on the endosome? Does the GPCR butt into a multivesicular body? What happens?
To arrestin.
What happens to the whole thing? Is it behaving just like any vesicle, that upon release from its donor compartment, the coat proteins are released? Yes.
Can you just repeat the question?
Oh, that's really hard. Let's see what we can do here. So the question is, how does this all work? That's the real question, right? So if arrestin binds, right, so arrestin binds and recruits clathrin. So the normal model for protein-coated vesicle formation is that adaptin and clathrin are released after the vesicle or endosome buds off, right? But that leaves a question that Austin had, which is, well, would this butt into a multivesicular body? And I said, well, maybe not if arrestin is still bound. But then there came this question, which was, well, if arrestin is still bound, then that's not following the model of how adaptin and plasmin would be released. So really, what's going on here? Hopefully you can follow. So you guys are asking for a level of detail I do not have, but I'm sure people have said it. Oh. That was fun, guys. I love it. You know what? I love it when we stay on a topic for a long period of time because it allows me to see how you guys understand things. And it might be frustrating for people who just want to keep moving, but I'd always rather you learn 2 things really well than 100 things not very well at all. So. Okay, so let's move on to signaling through enzyme-linked / coupled cell surface receptors. Right, so there's 6 classes. Don't memorize these. Receptor tyrosine kinases, tyrosine kinase-associated receptors, receptor serine threonine kinases, histidine kinase-associated receptors, receptor guanyl cyclases, receptor-like tyrosine, basically enzymes that are linked to or enzymes that are either bound to receptors or receptors that have intrinsic enzyme activity. So you could almost put it into 2 classes, right? Either they bind to an enzyme, often a kinase, but here you've got guanyl cyclase, tyrosine phosphatase, or receptors that have intrinsic kinase activity. What does intrinsic kinase activity mean? Part of the protein, right? So let's talk about receptor tyrosine kinases. So these proteins are going to have, obviously they all have a transmembrane domain, and they all have a tyrosine kinase domain. Does it make sense that the tyrosine kinase domain is on the cytoplasmic side? Why isn't the tyrosine kinase domain on the extracellular side of the plasma membrane? This is a silly question. Why isn't the tyrosine kinase domain on the extracellular side of the plasma membrane? Yep. Right, you use it for phosphorylation, and more importantly, phosphorylation that results in signaling. So if it's outside of the cell, are you going to be able to signal anything to anywhere inside the cell? No, because you've got this darn membrane in the way. And so let's go back for a minute to when we were talking about translation of proteins into the ER, and the membrane proteins, and the transmembrane domain, and that was like the N-terminus and the C-terminus, and the topology, and this and that. This is really important that we have the right topology. Well, because you want to make sure your kinase domain is inside the cell. Because if it's outside the cell, well, then that's not a very functional receptor. And so then on the outside of the cell, you have your different ligand binding domains. But the thing that makes RTKs is they all have a tyrosine kinase domain on the in the cytoplasmic tail. Okay. So this is intrinsic kinase activity, right? Because there's no kinase bound to it. It's a domain within the receptor. So it is a bit of that receptor that has the kinase activity. Does everyone understand that? Mm-hmm. Raise your hand if you don't, and I'll say it again. Thank you. Yeah, so what I try to always differentiate between receptors that have intrinsic kinase activity and receptors that bind to kinases and use the kinase activity from a bound kinase. And this is an example of intrinsic kinase activity because these proteins have a kinase domain. They have a bit of their cytoplasmic tail has the ability to phosphorylate things. Okay? Good. So when the ligand binds to these types of receptors, often there's multiple receptors, and they need to dimerize. And the ligand binding, it actually brings the receptors closer together. So you can see there's, in this case, the ligand is a dimer, brings the receptors close together, and so then you get cross-linking of the receptors, so they're brought close together, and phosphorylation. It activates their kinase domain, and they can phosphorylate each other. And so this is 3 different receptors showing the same thing is happening, where you've got the ligand bringing the receptors together, then the kinase domains get activated, and they can be phosphorylated, and they can phosphorylate each other. Here's an example. This is a soluble ligand, right? But here, this is an example of just kind of like what we talked about with CD4, where you don't have CD4 binding to a soluble ligand. You have CD4 binding to a receptor on another cell. So these are 2 cells that are signaling to each other and communicating to each other. And so one has a cell surface protein that's acting as a ligand for the receptors. Acting the same cell though, cross-linking them, bringing them close together, activating their kinase domain so they can phosphorylate each other. What's the purpose of that phosphorylation? Yes?
To create binding sites?
Right, to create binding sites. Because then you're going to get all these signaling complexes recruited. Remember on Tuesday, I talked about how different signaling complexes can be recruited to phosphorylated receptors. So that's what would happen. Okay. You're studying, here's a practice problem. You're studying a new receptor. It's always new, isn't it? Is it worth studying an old receptor? I guess as long as you're finding out something new. Nobody cares if you're just doing something someone else did before. Huh? Oh, okay. So unless you feel like the original work was falsified or plagiarized, then you should probably study the old receptor. But then you're a whistleblower, and no one likes a whistleblower. I'm just saying, even if you feel like you are in the right, your career is going to be destroyed. Anyways, I'm just saying. I'm not saying you should keep quiet, but I'm saying, like, figure out a way to be real nice about blowing that whistle. You are studying a new receptor, not a falsified or plagiarized one, and you believe it is involved in regulating expression of genes associated with cytokine secretion, so remember, expression, production, in the presence of gram-negative bacteria. So this is an immune response, right? Design an experiment to determine whether a receptor functions in this signaling pathway. So you have to think to yourself, how is this receptor activated? Can I mutate something? Can I, what would be my readout? What would I look for to see if the receptor is active? Think about those things as you discuss with your friends or friends. Maybe they're more than one friend. I'll circulate if you have questions about how you're doing my things. Who said what's a cytokine? A cytokine is a secreted protein. So it's a signaling molecule. So it would act as a ligand for your receptor. Well, in this case, it's the response. It's acting with the response. So it's an expression. Who's ready? Okay, that was a long one. I'm sorry about that. Hopefully you guys enjoyed talking about receptors. Those who didn't talk about receptors, hopefully you enjoyed having a brain break. I'm guessing you were all talking about receptors though. Yeah. No one was taking a brain break. Okay, so the real way to approach this is to think about what we just learned. Because I had someone coming up with wonderful ideas about GPCRs, but really we're talking about RTKs now, and so I wanted you to think about RTKs. But if on an exam, if I could specify, if you spoke about something that answered the question from a GPCR perspective, that would be fine too. Okay, so who wants to, actually I'm going to call on someone. Tamika. Just tell me, you can just tell me how you approached it, or you can give me the answer, whatever you feel good about. That's perfect. That's a perfect answer. So what Tamika was saying was what she first thought about was that something needs to go to the nucleus to activate gene expression to get cytokine secretion. So that's your readout, is to look for cytokine secretion. And that you know that the receptor needs to become activated in order for this whole signaling pathway to make that happen. So you need to start to think about, well, what activates the receptor? And so you can either do what a lot of people call the oversight, which is delete the receptor. Just delete the whole receptor from the genome. See if when you add bacteria, obviously you would have wild type cells, and deletion mutants, add bacteria to your cells and look to see if cytokines are made, or if the gene is expressed with the RNA, with the protein, whatever you want to do. Yes, you can do that, and that would not be wrong, but that was not what I was looking for. But what I wanted, which is what Tamika just said, which is you can also delete your kinase domain, because that kinase domain is required for activation of signaling. So you may have wild type cells with normal receptor, have mutant cells with the kinase domain removed, add your bacteria, look for cytokine secretion. And so that was it. In all reality, you have to do it all. I also got an idea from the back that was, well, can you make your bacteria fluorescent and see whether or not it binds to the receptor? Yes, but that would answer whether or not your bacteria binds to your receptor, not whether or not it activates gene expression. So, but in the paper, you would do all of these things because you have to look at every single part. Yeah.
Instead of deleting that whole region, could you just mutate it so that it doesn't function correctly?
Yeah. So the question was, instead of deleting the whole domain, can you just mutate it so it didn't work? Yes. So like every enzyme, there's an active site. And so what you usually do is a single point mutation, where you take that active site, which is usually cysteine, stuff like that. You take that active site, you make a mutation there so it doesn't work. And actually, you would want to do that rather than delete a whole domain, because when you delete large chunks of a protein, you're going to encourage misfolding. And then that receptor might never make it to the cell surface for you to even do your experiment because it'll be degraded by URAD. Oh my God, she said URAD, signaling. Questions, thoughts? Who had other ideas that they want to share? Yes? Right, exactly. So that's kind of what Tamika was saying is like you can cut off the domain, you can delete the whole protein, or you can cut off the cytoplasmic tail. So they're all different levels of mutation. But you always, for those of you who are going to work in the lab one day, you always want to be very conservative when making mutations so as not to destroy your protein structure. Because when your structure is destroyed, then nobody believes anything you do. Yeah?
I'm just wondering, how do you actually cut this stuff off? Is that like
It's all in the cloning. So how do you cut off parts of a protein? So have you worked in a lab ever? A little bit? Have you done PCR? Okay, so usually, everyone's learned about PCR, where you make your primers and you amplify your gene of interest, right? And so that's also how you can make mutations. So you can do multiple things. If you're doing a point mutation, here I'm going to draw. Okay, so here's your gene. Woo! There's your gene of interest, okay? And so you know your sequence. And so what you can do is you can design overlapping primers. So you would design a primer here that goes this way and a primer here that goes this way. Let's say you want to make your mutation right here. And you want to make a cysteine to an alanine. And you know the codons for cysteine and you know the codons for alanine. And so when you make your primers, you literally call up the company, actually use a website. You go to the website and you insert the sequence of the primers you want them to make. And then they send them to you. And so you just put in your forward and reverse of primers that mostly match your template. So they're mostly going to match your gene of interest, except for they're going to have one or two bases that are different that are going to, upon translation, create your alanine. That's for a point mutation. If you wanted to do a truncation, you can insert a stop codon, right? Because that would just stop translation. If you wanted to cut out a huge chunk, you could also, if you have, so you'd have it in your plasmid, right? That's how you express it. And if you have it in a plasmid, your gene would be here and here. If you wanted to cut out a big chunk of your gene, right, you can look for restriction enzyme sites that would cut it out, right? So you might look for an enzyme site that would cut this part out if you know what, you would know what you're targeting. And then you would end up with that, it would ligate back together with that part missing. And so then you have this plasmid that has your mutant in it, and you express it in your cells that you're working with and see how it behaves. So in this case, you would take this plasmid and you would put it into your mammalian cells. You would throw your bacteria on it, and then you would either isolate RNA to look for production of the RNA for that cytokine, or days later you would take the media and look for presence of the cytokine. So that's how you would do that. These are all fun things you get to learn when you work in the lab. Any other questions about that? This is why it's so important to have a lab component to a class like this, but unfortunately, well actually there's a cell lab starting next semester. Yes. Yeah. Are you graduating? So stay. Stay. It's supposed to be very exciting. I think I'm going to be helping out a little. Okay. 5 minutes left. Okay, so we're talking about kinase domains, right? And we're talking about how there's all this autophosphorylation, phosphorylation of self, right? Autophosphorylation of the cytoplasmic tails activates other kinases and creates docking sites for other intracellular signaling proteins that form a signaling complex. And now you've got all these phosphate groups hanging in your cytoplasmic tail, and it's going to create binding sites for all these other proteins that are going to help in relaying the signal through the pathway. And so, just like what we spoke about, if you wanted to inhibit signaling through this receptor, you could also, what would you do? So we've already talked about mutating a kinase domain, but now let's say you know all the phosphorylation sites. What could you mutate to inhibit signaling? Yeah, you can mutate any of those phosphorylation sites because that signaling complex is required to relay the signal, and so signaling actually happens. As soon as you mutate phosphorylation sites, you're not going to be able to reproduce proteins, you're not going to be able to activate kinase activity, all of that. Okay, so we'll stop here and we'll finish on Tuesday.